it's this, basically. So there are parts of your genome that are almost always heterochromatin. And those would be things like centromeres and telomeres. They never really open up. They're always somewhat condensed. The rest can go between euchromatin and heterochromatin, and that transition is essentially mediated by processes like this. And so heterochromatin, you have to be a little bit careful because some people sort of will use that term to mean really just the centromeres and telomeres because anything else can become euchromatic. But the more common usage would be to say this is heterochromatin and this is euchromatin. You're going back and forth dynamically between those two states. Centromeres don't really ever open up like this, as far as I know. Yeah. Right. Okay. So this is a really good question. Are DNA origins of replication the same as where RNA polymerase sits down? And the answer is no. The origins of replication for DNA polymerase are to get the whole genome copied. The places where RNA polymerase starts are just to make one copy of one gene at a time. Typically, I mean, in your cells anyway. And so that's done in a very different way. Each gene has its own start site. Again, bacteria, a little bit special case, we'll talk about that later, but just think about your genome for now. Each gene has its own start site, that's where RNA polymerase sits down, and instead of making a copy of the DNA, it's going to make a copy into RNA. So those two processes are regulated differently, they have different purposes, and physically they start in different places. And so again, we're going to spend a bunch of time talking about transcription, and then we're going to talk about DNA replication. So it'll start to make a lot more sense as we get into those topics in detail. Yeah. So we call it open and closed structure here because when the DNA is in this configuration, the chromatin is in this configuration, it's possible for that gene to be expressed. And it's open in the sense that not only does it physically look more open, you've got space between beads, but also because proteins can come in and do things on the DNA, like start up expression of a gene and make an RNA. So RNA polymerase can access the DNA because it's more open, more accessible. So it's open in two senses, physically and in terms of interactions with proteins that regulate gene expression. These states of configuration really are related only to mitosis and meiosis. Most of the DNA in the cell is like this. The actively transcribed parts are like this. Almost nothing's like this. And this really happens when you make a condensed chromosome for either mitosis or meiosis. Yeah. Yes, it would, if you think about it, because you're in the same length of space here, you have a lot more DNA wound around that chromosome. So that length and that length is about the same, but you've got way more DNA pulled in there. So the total length of the molecule, so your chromosome 1 is about this big. Okay, well, a little bit smaller, about that big. If it's completely naked DNA. But when it's like that, it's only about 5,000 nanometers. I can't even hold my fingers that close. So the amount of condensation has a huge impact on the total length of the molecule. You're essentially winding it up really, really tightly so that you can move it around without breaking it. You don't want to break that. That's a big, big molecule. You do not want to break it. The cell freaks out. Steve mentioned this earlier. A cell does not want to see naked, broken DNA ends. It will immediately try to repair them. And that's a bad, bad thing for you. And so we'll talk about DNA repair mechanisms later, but a lot of the purpose of doing all of this condensation is to make sure that that these are two single DNA molecules, that they do not break during the process of movement into the separate daughter cells during mitosis or question is about telomeres and getting shorter with time, with aging. So we're going to, what Steve introduced on Wednesday was the structure of the telomere. He didn't talk to you about how it's replicated. Okay, so we'll get to that when we talk about DNA replication. But you can see, if you think about it, that this is going to need maybe some special ways to get replicated that are different from the rest of the chromosome. So when DNA polymerase comes along here making copies to make the new DNA that's replicated into new molecules, it's going to have to do something special at this end. So let's not worry about what those mechanisms are, because we're going to talk about them in detail later. But the important thing to carry away from this is that unless you use telomerase, that special enzyme he mentioned, this end's going to get shorter and shorter and shorter. So if you look at a blow up of the ends, there's a repeat unit, TPAGGG, 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 and then you've got an overhang. This repeat usually goes back hundreds of copies or dozens of copies. Without the telomerase enzyme, you cut back every time you replicate. And they get shorter and shorter. Eventually, you're done with the telomere repeats and you hit regular DNA without those repeats, and you can't replicate the enzyme anymore. That chromosome cannot be replicated. The cell that contains it is what we call terminally differentiated. It will never make another cell. Most of the cells in your muscles, your brain, a lot of the parts of your body are of that kind. They will never divide again. When you build up muscle mass, you're building thicker fibers. You're not making more muscle cells. Your brain, there's very, very little neuronal cell division going on. There are other parts of your body where there's very active cell division going on, like your bone marrow and the lining of your stomach and intestine. Those are very actively dividing cells. They make telomerase, and it keeps adding more of these repeat sequences on, so you can continue to divide. That's the level we want you to know about it now. As to how the mechanism of replication happens, we'll talk about that. Okay, so there's a lot of interest in telomerase as an anti-aging component, as Steve mentioned, but that's a false thing. Those cells are not supposed to divide anymore. You're not going to become youthful just by allowing your muscle cells to continue dividing. Sadly. Okay. These are two options, and my understanding of this is that we don't know which of them is true. What we know is that it's a heterodimer. Remember, heterodimer means you have two partners and they're different from each other. In this case, we know these coil regions fold into long coils, but whether they fold back on themselves into a hairpin, or whether they intercoil, my understanding is that that's not known. The question is about how the histones come together to form a nucleosome. So in a nucleosome, there are how many single protein molecules are in a nucleosome? Anybody remember? 8. Okay, and there's 4 different kinds. You have 2 of each. So the first step is that 3 and 4 make a heterodimer, and then that pairs with another one of itself. So you really have a tetramer with 2 of each. And on the other side, you have 2A and 2B, and they make a dimer, and you have 2 of these, and so you have that tetramer and 2 of these dimers, and that together is going to give you the histone, and then the DNA wraps around that. But it's this tetramer that nucleates the wrapping process. And this is a dynamic process. These histones the octamer can come together and go apart, and come together and go apart, can get assembled and taken apart. When the DNA polymerase comes through here to replicate DNA, or when RNA polymerase comes through to make messenger RNA, it will pull will disassemble that structure. So you will actually go, you will actually transiently go into naked DNA, and then you'll have to put this back together. That's just when the enzyme moves across them. Generally speaking, that's the only time. Okay, so this is a really good question. The question is that when the DNA wraps around the histones, is there a specific sequence in the DNA that interacts with the proteins? We talked about restriction enzymes being specific sequences. And people spent actually quite a lot of time looking for this. It was very clear early on that there was no highly specific and unique sequence. But there was sort of this thought that maybe there would be something fuzzier, like you need at least 2 of these in one of those, and 5 of these in one of those. And it turns out that there's not a lot of sequence specificity that goes into this process. That the spacing of the beads on the string is roughly the same, but I'm going to show you a picture of the beads on a string. I can find it. Okay, so this is a micrograph. This is an electron micrograph of actual histone octamers in that beads on a string configuration. And I think what you can see is that the spacing is not perfectly even. Now some of that could just be the molecules a little bit kinked up and it's not stretched out perfectly. But even where it's nicely stretched out, you can see that it's not exactly even. Sometimes you've got a little more space, a little bit less space. The short answer is that spacing is fairly regular. It doesn't seem to be highly sequence specific, and that there isn't any requirement for certain kinds of sequences to get the beads on the string configuration. That kind of makes sense because you need to be able to do this everywhere. You don't only want to make the histones attaching in certain places. You need to be able to make the beads on the string throughout the chromosome, or you wouldn't be able to condense it into the 30 nanometer fiber and so on up into the higher order condensations to be able to execute mitosis. So that's the long answer to it. It's a very good question because there was a lot of concern that that might be actually part of how gene regulation happens. It may still be a subtle part of it, but it's not a really obvious part. So this space in here is really just tens of base pairs. It's too small to hold a gene. Now, Steve didn't talk about it, but there's a fifth kind of histone called H1. You'll notice we've done 2A, 2B, 3, and 4. H1 is actually responsible for binding in that space in there and pulling these guys together. And there are different H1s that give you different amounts of compaction and things like that. But so let's not worry too much about that. But the only thing you really need to know is that that's way too small to hold. Yeah. Right. What this is. Isn't that practical? So right here. Okay. So actually what you're looking at here is the 30 nanometer fiber. It's not, so this denser one is this, and that, the real loops that come out are actually less condensed. So you'll notice that's thicker than that part. And then even in here, that opens up into the beads on the string. So this was first noticed in what were called lamp brush chromosomes. This is a historical background, you know this. Where you could actually see that the chromatin had thicker and thinner regions, and the loops were known to be the place where genes were actively being transcribed. And that was one of the first forms of evidence that the state of condensation mattered for gene expression. Is this with reference to the histone tails or? So this phosphorylation is just one of the ways you can modify the histone tails and change the chromatin configuration and also affect which proteins come down and attach to the chromatin and then turn on and off genes, which is something we're going to be talking about much more in the next couple lectures. In the cell cycle or something like that, I mean, it's more look. One way to think about these modifications is they're very local micromanipulations of the chromatin. They're not something that's going to happen across the whole genome at once. You're going to be just in this place and just in that place, and there's going to be a lot of micromanagement because you're going to be turning on and off genes all over the genome. So this is a fine-scale control process. It's not something that's going to affect all the histone optimums at once. This one. So the question is this denatured DNA? So let's think about how you do DNA precipitations the way you do it. Would you be able to see this if you took the DNA you precipitated? So some people are shaking their head. Someone want to take a stab at the answer to this? Out loud. Just think through what the process we used for isolating DNA was. What chemicals did we put in there? And what did that do? What would you predict that would do to proteins attached to DNA? Yeah. So let's think about what alcohol on high salt would do to protein DNA interaction. Why do you think the DNA is sticking to that histone optimers and not around? What's causing it to do that? Pardon? There. Histones are positively charged. Okay, you remember that? That Steve said they're rich in lysine and arginine, which are positively charged amino acids. DNA is negatively charged, so they're binding together with what? They're bonding with the backbone, and those are what kind of bonds? Ionic bonds. So if you put a lot of salt in solution, what are you going to do? You're going to break up those ionic bonds. So the DNA is more or less going to pop off the histones in high salt. Okay. Now it's not, when you guys precipitated DNA in the lab, you weren't getting all the protein off. Some of it's just too sticky, but mostly you're getting a lot of the proteins off. So to get this kind of micrograph, you're going to have to isolate the DNA in a special way that preserves the ionic balance that is in a normal cell. So this is a very special preparation to be able to see something like this. You also have to be very, very careful not to break the DNA. These long, floppy molecules are really fragile. So this kind of, even though you sort of look at it and go, oh yeah, fine, these are the screen, that it actually is quite difficult to be able to get the conditions right to isolate the DNA with the histones on it to be able to see it. Yeah. Okay, so the question is, DNA is rarely in a naked form, but when the cell's not undergoing mitosis, what form is it usually in? And the answer is it's mostly in this 30 nanometer form, so it's a little more condensed. Again, I want to go back to you guys really need to think about DNA as a very physically fragile molecule. And a lot of the reason for condensation is just to keep it from getting broken. And so you're only going to open it up when you really need to. Supercompaction really only happens during mitosis and meiosis, but this is sort of, think of that as the normal habitat of DNA in a eukaryotic cell. Bacterial cells, they don't wrap it up this way. They use those other proteins, those SMC proteins, but in a eukaryotic cell, that's the normal configuration of DNA. You know, it's going to go to Steve's head if I tell him that you had no more questions about his lecture at this. I mean, seriously, he's going to be like, wow, he nailed the lecture. So you can't let that happen. Yeah.
So somatic cells don't produce telomerase, right? Somatic cells don't make telomerase, right? That's what he said.
They dial it down, yes, but basically that's true, yes.
Okay, but they still make some? Because I don't know. Could you just like go over that? Like, I don't know. It just
So
like
this is the reason for me being a little ambiguous about this is there's no hard and fast rule. I would hesitate to say all somatic cells make zero telomerase.
yeah.
Some somatic cells don't divide under normal conditions, but might under particular conditions like wounding or stress responses, so that they wouldn't make much telomerase but reserve the capability. There are others that absolutely cannot divide.
Yeah.
So it's not an absolute distinction. Chris had a comment to make.
Well, I was just going to point out that even if the cell isn't expressing telomerase, it still has the gene.
Right, it can turn it back on again, yes.
Right signal.
Yeah.
To get that gene on again.
Yeah. Some cells can reactivate a mitotic program if they need to, even though they wouldn't under normal circumstances.
Uh-huh.
So these are all reasons why we can't sort of give you an absolute answer to your question.
So like you get like new skin cells pretty often, right? Like you get new skin cells pretty often.
Yeah.
Is that because your skin cells are expressing telomerase, or they're like, I don't know.
Right, so this is an interesting question, and the general answer, not just for skin, but for a lot of places in your body, is that there's continual renewal of the cell population, but it's coming mostly from stem cells dividing.
Okay.
And so stem cells are making plenty of telomerase, and what stem cells do is they make two kinds of divisions. One is they make more stem cells, and the second is sometimes they divide and they make another stem cell, but the other cells can become differentiated. That cell will then go on to actually become a fully differentiated skin cell, and you turn over all of your skin cells on, I can't remember, it's like a bi-monthly basis or something like that. Every single cell that you call skin, you didn't have last summer, and this summer you'll have none of the ones you have now. There's always renewal going. The same is true of the lining of your gut. The same is true of your red blood cells, many of your white blood cells. There are tissues in your body that are constantly renewing, and then there are tissues in your body that are static, like skeletal muscle and brain. Okay, so there's a wide, and there's everything in between. So that's why there really isn't one answer about this. But it's as you can imagine, intensely important for thinking about things like cancer, wound healing, autoimmune disease, all these things, these kinds of capabilities come into play. Which is why people care a lot about telomerase.
Mm-hmm.
Yeah. That just means no proteins on it. When we say naked DNA, we mean no proteins attached. Right. So again, it's one of these things. If I show you this picture, I'll say that's not naked DNA, but actually, as far as those base pairs are concerned, it is naked. I mean, so it's and when the polymerase moves through to make more DNA, of course, the polymerase is there then. But right in front of it and right behind it, there's a little piece that's not got anything on it. So it's not literally true that a single base always is touching the protein, but regions always have something attached, whether it's the polymerase or histones or other kinds of interactions. There's almost always something going on. It's very rare to be true, like an extended region of truly, more than a couple bases. It would be a short region, like the string between the beads. Would be an example. That's pretty close to naked DNA. But it's short. Yeah. Right, so this is a great comment. So you ask as a question, but you're stating it as it's actually true. So the question/comment was, during development, when cells are differentiating, the regions of the genome in one cell type versus another cell type that are heterochromatic and euchromatic, they can be different. Right. Right. Okay. So I want to, because it's such a great point, I want to first address the point. So do people understand why that might be true? Why would you expect in two different cell types, the part of the genome that's heterochromatic and euchromatic to be different? Think about why you go back and forth between these two states. Why did I say, what would be the reason for a cell to change the configuration in a particular region of the genome? Expressing different genes, okay? Different cell types express different genes. So it's going to modulate that configuration change differently in different cell types because those different cell types are expressing different genes. A muscle cell and a brain cell are going to make very different sets of genes because they functionally do very different things. So it's going to micromanage the chromatin differently in those two cell types. But all muscle cells are going to do it the same way, so it's approximation. And brain's a little more heterogeneous, but different kinds of neurons are going to roughly do it the same way because they're going to use some of the same genes. And those are going to be really different. Okay? How does the cell do that? It does it through these modifications. There are a set of enzymes that mediate these processes, and now we're getting into regulation. Again, we're getting a little bit ahead because we're going to mostly be talking about this in the next couple of days. But there are sets of proteins whose job is to either add these covalent modifications or to remove them. So kinases add phosphate groups to other proteins, and phosphatases take them off. There are acetylases that add acetyl groups, and in fact there's a dedicated set of histone acetylases and histone deacetylases, and their job is to add these acetyl groups or to take them off of the tails to do the modifications. But then your question is going to be, well, what tells them whether to do that or not? Signals that the cell receives at the surface, changes in cell physiology, developmental switches, all of these things feed into the activity of the histone deacetylases and acetylases, the histone kinases and phosphatases, ubiquitinases, methylases, all these, there's a whole dedicated set of proteins that are doing this stuff, and they're receiving input from signals elsewhere in development in the cell and the environment, the immune system. All those are feeding information in and changing the activity of these proteins. So they're, in a sense, they're kind of gatekeepers of chromatin configuration, but they're getting their instructions from other processes in the cell. Well, there are. I mean, those are proteins that regulate, but they're receiving instructions also from other proteins. So it's not, almost no protein is going to function on its own regulation. It's part of a network of interactions. And it's the structure of the interactions that determine the regulatory outcome. Yeah. Why it switches back and forth? Okay. You can't make RNA when it's compacted like that. So it opens up when a gene needs to make RNA. So in order to get the RNA made to express the gene, you need to go from the 30 nanometer fiber to the desome string. And that opens up the DNA a little bit more so that the enzyme can come, the RNA polymerase enzyme can come in and make RNA. None of these could you make RNA from a gene. You have to be opened up at least that far. And you go back here to make sure you don't make it at the wrong time. So we're not only interested in on switches, we're also interested in off switches. So you don't want to make hemoglobin in a muscle cell because that's not appropriate for that cell. So the region around those genes will be wound up like this in a muscle cell. On the other hand, it will be open around the genes that are appropriate for that cell. These are for cell division, mitosis and meiosis. I mean, there is some of this going on too, but these really condensed versions are really for cell division. So we haven't explicitly talked about gene regulation yet. I'm going to talk about that on Monday. and sort of set up, and really next Wednesday we'll really start getting into it. But the next 4 lectures we're going to talk about gene expression. And particularly in the last 3 of those 4, we're going to be talking about nothing but regulation. So a lot of this will start to come together. Okay. It's a dynamic process, so don't think of one part of the genome as permanently heterochromatic or euchromatic, except for the centromeres, which are almost all the time heterochromatic. But most of the rest of the chromosome can switch back and forth between the 2 states. So we're going to call it euchromatic when it's open and heterochromatic when it's closed. A particular region might be heterochromatic in one cell or euchromatic in another cell, because in one cell you don't want that gene on, so you're going to make it heterochromatic. And in another cell you want that gene on, so you make it euchromatic. Yeah. So it's part of the regulation process. Part of the gene regulation process. Correct. Okay, so this is a great question, and you're thinking ahead to exactly the processes we're going to be talking about next week. So the question is, if you can't fit a gene in there, do you have to open this up even more in order to get transcription to occur, or replication? And the answer is yes. So there are, on top of this alone is not enough to turn a gene on. But it allows the next step to happen, which is that additional proteins come in there, and then they open it up even more, and they help the RNA polymerase come in and attach. The RNA polymerase by itself will not attach to DNA that looks like this. It needs help. And those are additional regulatory proteins, which we'll talk about next week, but they're called transcription factors and sigma factors, proteins like that. There's a whole range of those proteins whose job is dedicated exclusively to helping RNA polymerase know where and when to turn a gene on. Yeah, it's a great question. You guys are thinking about this in a good way. You're racing ahead a little bit. I like that. But again, like we so often do, ask you to be a little patient until we get into that stuff in more detail. But that's exactly the right kind of way to think about these problems. It's not denatured in the sense of when we talk about denatured for DNA, we usually mean single-stranded. You're thinking dissociated from protein. Okay. So it's not denatured in the sense of being melted or single-stranded. It absolutely is. Right. So the question is relating to the function of restriction enzymes in the natural context and whether histones get in their way. If you think about where restriction enzymes function normally, their context is in a bacterial cell. So there are proteins on the DNA, but they're not histones. Okay? So they're the SMC proteins. So those proteins aren't as tightly wound around the DNA as this. And so remember that restriction enzymes are the bacterial cell's analog of an immune system. They're there to identify foreign DNA and chop it up so it can't function properly and infect the host cell. So that foreign DNA typically is going to be coming in either naked or very close to it. That's very different from the way DNA exists in your cells. So it's just a different situation. Plasmid DNA, viral DNA, which are invaders as far as the bacterial cell is concerned. At least plasmid DNA sometimes can coexist with sometimes, but certainly viral DNA coming in. It's an invading DNA, and you want to get rid of it. And one of the characteristics that the cell uses to do that is the fact that it can recognize the specific base sequences and chop them up. So I don't know if there are any eukaryotic cells that make restriction enzymes. You can express it, but it's not naturally there, right? Yeah. because of the isol, the way you isolate it, stripped off 99% of proteins. There's still a little bit of protein on it. I wish we could get DNA completely clean of protein, but the truth is it's always got a little bit on it. I'm sorry, could you rewind? Okay, so the question's about the specific structure of the telomere and the T loop and the D loop. This is going to make way more sense to you when you see how it's replicated. So for right now, really all we want you to know is that it's a special configuration, that there's an overhang, so there's a 3' overhang, and that this ties it up in a nice little bundle so that there's not a naked end. There's not a free 3' and 5' end for the exonucleases to see. Okay? This hides the ends away so that the enzymatic machine can't see it anymore. It's like, if you've ever worked with knots, you do that thing at the end of the rope where you kind of make it not frayed and stuff like that. That's sort of what the cell is doing here. It's keeping it from unraveling and getting digested by exonucleases. This structure has to do with the way it's replicated. And so let's just save that for when we talk about replication. Otherwise, I'm going to have to go through the whole process now in the slides, and it's not going to make a lot of sense. But all of this is repeat. This is all telomere repeat. It's not until you get out here that you're in the regular chromosome territory. So it's not like the repeats are just at this part. It's this whole area is telomere repeats. Okay, so these are the microtubules. There's a bunch of proteins out here and here that's a mix of proteins. And they include proteins that interact directly with the microtubules, proteins that interact directly with histones, and proteins that link all those together. So it's a complicated mix of proteins, but they're essentially allowing attachment of the microtubules to the chromosomes via these intermediary proteins. Anything else? Enjoy the beautiful day. Have a great weekend. We'll see you on Monday. They are less